Welcome to Biomedical Engineering Faculty in Focus. This is a series we highlighting the fantastic research our faculty are performing at the Department of Biomedical Engineering at Columbia University. My name is Edward Guo. I'm the chair of the Department of Biomedical Engineering here at Columbia, and welcome to join us in the BME Faculty in Focus. And today, it is really truly my pleasure to welcome Professor Wang to present really exciting, outstanding research in the neuroengineering. And so he has done very innovative work in looking at neuromodulation of neural coding and decision making. And that's all yours.
Thank you very much for introduction. Let me share my screen here. Okay. Yeah, it's a great pleasure to be here. Thank you, Ed, for nice introduction. Yeah, I'm very excited to share some work we have been doing in the past several years and to tell you some ongoing projects. So first, required by Columbia Financial Conflict Interest Committee, I have to disclose my potential financial interest. I'm co-founder and have financial interest in Sharper Sense, the company I co-founded with my student. Right. So for my work, we all understand sensory signals such as photons or pressure wave or chemicals or mechanical deformation on the skin are transduced to neural signals or spikes at the periphery. And these neural signals are transmitted to the brain through several stages pathway to form the perception to guide our behavior. So my lab is fundamentally interested in two big problems. First is how is sensory information processed in the brain to form perception to guide our behavior, and how is this process modulated by different neuromodulatory systems. The second problem that we are interested in is more from engineering perspective, which is how to interface with the nerve system to restore or enhance information processing in the brain. So the first story I like to tell you is about Lucas modulation of sensory processing. So why Lucas? So Lucas is a small adrenergic nucleus in the brainstem. It's the only source of norepinephrine, which is a very important neurotransmitter to the forebrain. And the Lucas norepinephrine system is a major neuromodulatory system. And the malfunction of this system has been implicated in many major neurological disorders, including Alzheimer's disease, depression, ADHD, and schizophrenia. So our first question is how Lucas modulates feature selectivity in the thalamus. So what's a feature selectivity? So in early stage of sensory pathway, sensory neurons are typically sensitive to a certain feature in the stimulus. That's technically their job. So this is called feature selectivity. The feature selectivity can be estimated by using linear, nonlinear Poisson model and reverse correlation analysis. So in this model, the first stage or component is a linear filter or the feature this neuron encodes to or sensitive to. The second component is a nonlinear tuning function that maps filter output to instantaneous fine rate. So in here, filter output is a convolution between stimulus and the feature this neuron encodes or filter. So more similar the stimulus to this encoded feature, the higher or larger the filter output. So in here, you can see this nonlinear tuning function also indicates how selective this neuron is to this encoded feature. Why is that? Assuming this curve is flat, assume this curve is flat. That means this neuron is not sensitive or selective to this feature at all. Why? Because its response to this feature is the same as response to any random feature. So in our experiments, we provide the white noise sensory stimulus to the neuron, and we simultaneously record the spiking activity from this neuron. So in here, we assume the stimulus right before each spike is somewhat similar to the feature is encoded. Therefore, we just copy paste all the stimulus right before each spike. All these segments of the stimulus called spike triggered ensembles. By analyzing the statistical structure of these spike triggered ensembles, we can recover the feature selectivity of this neuron. So to study Lucas modulation of thalamic feature selectivity, we recorded single unit activity from the ventral posterior medial nucleus of the thalamus in response to whisker stimulation while systematically stimulating the Lucas. There are three levels of stimulation. So first is a control, which means low stimulus. The second is 2 Hertz LC stimulation. The third one is 5 Hertz LC stimulation. In here, the ventral posterior medial nucleus is a thalamic relay stage of the whisker system. So in here, I'm going to always call it VPM. So before we microstimulate LC, we use electrophysiology to identify Lucas. So in here, they are very straightforward to identify if a neuron is a Lucas neuron, because Lucas neuron has 3 hallmarks. First, they have very, give me a second. Let me stop here. Yeah, they have a very wide waveform. This is a single unit activity that we recorded from LC. They have very wide waveform. This is a recording system. This window is 1.2 milliseconds, but this is only half of their spike waveform. So their full spike waveform is more like in here. So this is typically around 2 to 3 milliseconds as compared to 1 millisecond with a waveform of regular spikes. So second, Lucas neuron respond physically to pull and tail pinch. Third, there's auto inhibition following this phasic response. Therefore, it's very straightforward to tell if a neuron is an LC neuron or not. Of course, in some experiments, we also use histology to confirm correct placement of recording electrodes in the Lucas after recording. So for VPM, it's relatively straightforward to tell if a VPM neuron, or if a neuron, VPM neuron, because they respond furiously to the whisker stimulation, and the latency of their response is typically very short, is around 5 to 8 milliseconds. Of course, in some experiments, we also use histology to confirm correct electrodes placement in VPM after experiment. So once we find VPM neuron and the LC neuron, we switch the LC electrodes to microstimulator to microstimulate to the LC while simultaneously recording single unit activity from VPM. And from this recorded activity, we can estimate the feature selectivity of these VPM neurons. So first thing we found, LC stimulation significantly decreased the firing of the VPM neuron to the same stimulus. So this is a plot plotted in control versus LC stimulation, and there are 2 conditions. Purple is 2 Hertz LC stimulation, and orange means 5 Hertz LC stimulation. And each dot is a VPM neuron. As you can see, almost all dots are below this unity line, which means they have a higher firing rate during control as compared to LC stimulation conditions. However, to our surprise, we found LC stimulation significantly increase the feature this neuron encodes. This is again the plot of example feature encoded by the same neuron with and without LC stimulation. As you can see here, blue trace is a control condition, which means no LC stimulation. Purple is 2 Hz LC stimulation condition, and the orange is 5 Hz LC stimulation. So you can see 5 Hz LC stimulation significantly increase the amplitude of encoded feature while maintain the shape to be the same. So to quantify this increase in encoded feature amplitude, we calculated feature modulation factor here. So in here, feature modulation factor is a dot product between conditional feature and control feature normalized by dot product of control feature and itself. So if conditional feature is about the same as a control feature, we should expect a feature modulation factor of 1. However, what we found is not the case. 2 Hz LC stimulation increase the amplitude of the encoded feature by around 20%, while 5 Hz LC stimulation increase the amplitude of encoded feature by 50%. So we also find LC stimulation make this neuron more selective to the feature it encodes. Why that? Because we found the nonlinear tuning function tilted up. So as I just said, the flatter this nonlinear tuning curve, the less selective this neuron to this encoded feature. But in here, this nonlinear tuning function tilted up. That means this neuron becomes more selective to this encoded feature. So now in here, we found LC stimulation has 2 beneficial effects. First, increase the amplitude of its encoded feature. Second, makes this neuron more selective to the encoded feature. So both are beneficial. So to collectively quantify this beneficial effect, we use the information theoretic approach to calculate the information transmitted by each spike, which is a probability of the encoded feature and the observance of this each spike. What we found is 2 Hz LC stimulation increase the information transmitted by each spike by about 250%, while 5 Hz LC stimulation increase the information transmission by about 450%. So we're very excited about this. But the one thing we need to know if this is artifacts of electrical microstimulation. So in here, as I just said, we use electrical microstimulation to activate the leukocytes. Because leukocytes is very small in rats, is about 300 microns by 300 microns. So it is possible this improvement that we observed in experiment due to current spread to adjacent nucleus to LC. So to control for this, we used optogenetics stimulation to activate LC. So in experiment, we inject lentivirus into the LC. Lentivirus carries channelrhodopsin gene and also is governed by LC specific promoter, which means they are most target LC neurons. So after 4 weeks, we performed experiment. Then we use histology to confirm the expression of channelrhodopsin in the LC. So in here, you can see this green is tyrosine hydroxylase, which is a signature of the LC neuron, and the red is M-cherry fluorescent protein, which is a reporter gene of our virus. So co-localization of green and red indicate the successful expression of channel-dopsin in LC neurons. So with optogenetic stimulation of LC, we found LC activation also decrease thalamic fine rate, increase the feature selectivity, and also information transmission. So this means what we observe is real, is not due to artifacts of electrical microstimulation. So now we try to understand the mechanism. So in here, we understand the Lucas projects to many other neuromodulatory systems. So what we observed here could be the direct release of norepinephrine by LC activation in thalamus, or could it be indirect effects by LC activates another neuromodulatory system and in turn activate enhance information transmission here, or a combination of both. So to test this, we use a phentolamine, which is alpha adrenergic blocker, to test if this is the direct effects from LC or indirect effects from LC activation. So in here, before injection of phentolamine, LC activation can increase feature selectivity for thalamic neurons and information transmission for these neurons. After injection of phentolamine, which means after alpha adrenergic receptor blocked, LC activation has low effects on thalamic feature selectivity and information transmission. So this means the increased information transmission in the thalamus is due to direct LC activation. So then we ask, what's the other mechanisms? So in here, in the whisker system, BPM, our thalamus receive feed-forward input from brainstem. So it's possible this improvement in information transmission by LC activation could be inherited from the brainstem nucleus. So to test this, we recorded from principal trigeminal nucleus in brainstem, or PRV here, which sends feed-forward input to VPM. So to our surprise, we find LC activation has no effects on PRV neurons' feature selectivity and information transmission. So this summary plot of PRV neuron that we recorded about their feature modulation factor and the information transmission with and without LC stimulation. As you can see, LC stimulation did not change or improve feature selectivity and the information transmission. So that means LC activation induced thalamic information transmission improvement was not inherited from brainstem inputs. So we also know cortex sent cortical thalamic projection back. So would this improvement was due to the cortical thalamic feedback? So in here to test this, we used mucimol to inactivate the barrel cortex. So in here, mucimol is a GABA antagonist, which means going to activate all GABA receptors to inactivate the cortical neuron. So before injection of mucimol, if we flip whisker, we can see a large local field potential in response to whisker deflection. So after injection of mucimol, there's no change in local field potential, which means whole cortical column was inactivated. However, with cortical inactivation, we found LC activation is able to increase the feature selectivity and the information transmission. So that means this improvement in thalamic information transmission by LC activation was not due to cortical thalamic feedback. So we were puzzled. So if not from cortical thalamic feedback, not from this feed-forward input, then the mechanism must be within the thalamus. So in thalamus, we know, in addition to this thalamic relay nucleus, there's another thalamic reticular nucleus, or TRN, which provides inhibitory control of the VPM relay nucleus. So we took a closer look at our thalamic spikes. We found LC activation also decrease the thalamic burst firing. So the burst firing is a small group of spikes with a very short interspike intervals, and also preceded by a dead time of 100 milliseconds. So to make things more interesting, we find LC activation not only suppress thalamic burst firing, this suppression is also inversely proportional to information transmission improvement. So in here, the x-axis is normalized changing the burst rate. The more negative means more suppression of thalamic burst firing, and the y-axis is normalized improvement in information transmission. As you can see, there's inverse relationship, which means the more suppression of burst firing, the higher the improvement, a large improvement in information transmission. So that means the mechanism is really due to the thalamic burst firing. We all know thalamic burst firing is an indicator of T-type calcium channels in thalamus, but why they are related, how they linked to LC activation. So in here, some previous work in Slide's work give us some hint. So when thalamic neuron is deeply hyperpolarized and the T-type calcium channel is deactivated, a small input will induce large calcium channel, calcium current, which in turn induce several sodium spikes. So in other words, once T-type calcium channel are deactivated, either preferred or non-preferred feature will induce the same thalamic response. So from information transmission perspective, this is not optimal. Why? Because it's no way you can tell which feature was present from the thalamic spikes. So to test this, we reasoning if this is the mechanism, then thalamic the LC activation would also suppress the burst firing for TRN neurons or thalamic reticular nucleus neurons, because thalamic reticular nucleus provides inhibitory control to VPM neuron to hyperpolarize VPM neurons. So to test this, again, we recorded from reticular nucleus of thalamus TRN while activating LC. As we expected, LC activation significantly decreased burst rate for TRN neurons while maintaining its firing rate. So to conclusively test or determine the role of T-type calcium channel in this LC activation-induced improvement in thalamic information transmission, we used ML218 or TTAP2 to block T-type calcium channels. Both well-tested T-type calcium channel antagonist. So before injection of this block T-type calcium blockers, LC activation is able to increase the thalamic feature selectivity and also information transmission. So after injection of this T-type channel blocker, LC activation had no effects on thalamic feature selectivity and the information transmission. So we waited for 5 hours after the blocker washed off, and LC activation start start improving the thalamic feature selectivity and the information transmission again. So this tells us LC activation enhances thalamic information transmission through regulation of T-type calcium channel. Okay, so then we use computational model to confirm our results. So in here, we built a network model. Both in this network model, we have a VPM neuron and a TRN neuron. VPM neuron provide excitatory input to TRN neuron, and TRN neuron inhibits VPM neuron and itself. So both VPM and the TRN neuron are integrating firing outburst model. So in here, we fit our experimentally recorded brainstem trigeminal nuclear spikes into this model. So for both model, they are governed by 3 membrane current and 4 membrane current, excuse me, and 3 synaptic current. And norepinephrine effects was modeled by decreasing the leak current and the increased efficacy of EPSC and iPSC. So in here, when we apply norepinephrine effects on both VPM neuron and the TRN neuron, we found the burst firing decreases. And the feature selectivity of this model neuron, VPM neuron, increase, and also information increase by 150%. Since this is a computational model, we can titrate effects of norepinephrine in only VPM or only TRN. So when we apply norepinephrine effects only in VPM, we find the increase information transmission by 100%, and applying norepinephrine only in TRN neuron can only increase information transmission by 50%. So which means norepinephrine effects in VPM neuron is more important than TRN neurons. Okay, so our last question in this project is, if the brain will take advantage of this improved information transmission by LC activation. So to test this, we have to use wake behaving animals. So in here, we train animal to do go, no-go discrimination task. So in here, in this paradigm, a stimulus is random assigned as go stimulus, which means animal must respond to this stimulus to get some water reward. Another stimulus is called a no-go stimulus, which means animal must withhold their response to this stimulus to avoid punishment. So in our experiment, 1 second after trial onset, either go stimulus or no-go stimulus will be presented. If go stimulus is presented and animal responded to this go stimulus within the window opportunity, this is a hit and animal will get some water reward. Otherwise, this is missed trial. When no-go stimulus was presented, if animal responded, this is the false alarm, which will result in typically 20 seconds timeout. Otherwise, this is a correct rejection. So after training, animal can do this type task very well. So this is a video showing our animal doing this type task. Oh, give a second. So in here, this is the head fixed animal, and we use the tactile organ, which is with a whisker. So this is a stimulator. This stimulator gonna move either up or down. So the rule is very simple. If whisker goes up, which is no go, this is go stimulus, animal must respond for sweet water reward come out this range. Otherwise, they should withhold their response. So after training, you can see animal can do this task very well. Okay, yeah, that was a goal stimulus. As you can see, animal can respond to goal stimulus and reject local stimulus. So in our experiment, our task is slightly harder than the one I just showed you. We have a one goal stimulus and a two low goal stimulus, which means goal stimulus in here is 8 Hertz whisker stimulation, and the low goal stimulus, and we have two local stimuli, 6 Hertz and 4 Hertz whisker stimulation. So as you can see, once animal is able to perform the task, we deliver optogenetic stimulation to the LC and random selector trials. LC stimulation increase the hit rate or decrease the false alarm rate. So both what we wanted. So to quantify these beneficial effects, we use perceptual sensitivity to measure their performance. And this is the well-accepted model or measure and the signal detection theory. So as we expected, LC activation significantly increase the perceptual sensitivity for these animals, which means the brain is able to take advantage the enhanced information transmission by LC activation. So now I'm going to switch gear to tell you several other stories. So one project we're doing is try to understand where the pupil link arousal comes from. So what that means, so you probably heard saying eyes are windows to the soul. Why is that? That's because when you're excited or you put a lot of mental effort, your pupil dilates, so your eye light up. So why did this happen? So there are a lot of theory, but there's hypothesis showing stating this pupil dilation is resulted from LC stimulation. So we decided to test this hypothesis by measuring pupil response to LC activation. And turns out this is the case. So when we stimulate the LC, and the pupil dramatically dilates. So this dilation is in a frequency dependent manner. Okay. So we also tested if another major neuromodulatory system or cholinergic system contributes to this pupil dilation. So this is a collaboration with Andrea Tobias group at Baylor. So in here, we use 2-photon imaging to image leukocytes axon in the cortex or basal forebrain axon in the cortex. So basal forebrain is a major nucleus of cholinergic system. So then we correlate the pupil size with activity of LC axon or basal forebrain axon, and we found there's a significant correlation. We also found if we use a first pupil derivative to correlate the LC axon and basal forebrain axon, they are also significantly correlation. So to further confirm this, we align the LC axon activity and basal forebrain axon activity to a canonical pupil oscillation cycle. We found there is correlation, and more importantly, both LC axon activity and the cholinergic axon basal forebrain axon activity leads the pupil dilation. So, and also LC axon activity leads basal forebrain axon activity, which means LC activation dilates pupil, maybe cholinergic axon or cholinergic system may dilate pupil slightly later. Okay, so once we figure out the pupil is a reliable indicator of both cholinergic system and adrenergic system. We try to understand their role in perceptual decision making. So again, we trained animal to perform this tactile go-logo perceptual decision task while we simultaneously measure their activity. So for this behavior paradigm, there are four behavior outcomes: hit, correct rejection, false alarm, and miss. We plotted pupil size or pupil dynamics around the stimulus onset for all behavior outcomes. And we found there's task evoked phase pupil dilation for all behavior outcomes, but they have different time course and amplitude and also shape. I'll tell you more about this in a minute. So, but we also find even the baseline pupil size, which is a pupil size right before stimulus onset, is also different for all these four behavior outcomes. Particularly, the pupil baseline size is much larger. On the trials, animal decided to take action than on trials when animal decide not to take action. So we then ask, would this pupil baseline size is informative enough to allow us to predict animals' behavior? So to do this, we constructed a Bayesian decoder to predict animals' behavior based on animal's pupil size. It turns out the baseline pupil size is informative enough, so our predictor, our Bayesian decoder, is able to predict animals' behavior with accuracy much higher than chance level. So more interestingly, the better the animal's behavior, the more accurate our predictor Bayesian decoder is. Right. So then we try to understand the relationship between the baseline pupil size and their hit rate and the false alarm rate. So first thing we found, there's inverted U-shaped relationship between hit rate and the baseline pupil size. However, the relationship between false alarm rate and the baseline pupil size is a monotonic increase. However, this translates to an inverted U-shape between perceptual sensitivity and the baseline pupil size, and this inverted U-shape relationship between decision criterion and the baseline pupil size. So now let's focus on this phasic or transient pupil dilation in response to task. As I just said, there are four behavior outcomes in this behavior paradigm: hit, correct rejection, false alarm, and the miss. And for each behavior outcome, the pupil dilation or phasic pupil dilation is totally different. We believe this is because each behavior outcome engages or involves different information processing component. For example, for hit trials, it engages stimulus encoding, decision formation, and motor execution to indicate this is a go stimulus, and also has the animal had some water reward or positive feedback. For correct rejection, yes, also engage stimulus encoding, decision formation, but also animal must withhold the response. So this is the response inhibition. So same thing for false alarm, engages stimulus encoding circuitry, decision making, motor execution, and also in here, since it's a false alarm, animal will trigger false alarm tone, they will selective feedback. For miss, is the same for stimulus encoding, decision formation, but animal decide not to lick, so must withhold their response. So We then use machine learning approach to decompose the kernel for each information transmission, processing component. So here our assumption is all these information processing components engage a canonical phasic dilation. We call this kernels. Then by using this machine learning technique, we can learn the kernels and also beta weight for each trial. So it turns out this method is very accurate. This is estimated kernels. By using these kernels and their beta weight, associated beta weight, we are able to reconstruct the pupil dilation at fairly accurately. So the advantage of using this method is it not only allow us to learn the kernel, also get a beta weight on trial by trial basis. Then we start to see if this beta weight is informative. So in here, we plotted beta weight associated with stimulus encoding kernel for go trials or no-go trials. We found these two distributions are different. More interestingly, if we calculate normalized distance between these two distributions, and this normalized difference between these two distributions are proportional or positively correlate with perceptual sensitivity. However, if we use the same analysis for beta weight associated with decision formation component, we did not find significant correlation. So which means this method is informative. So the last story I like to tell you guys is how we translate this knowledge we create in our lab. So as you I just told you, direct LC activation can increase information transmission in the brain, also improve animals' perceptual behavior. However, to direct stimulate leukocytes in the human is very challenging. This is because the location of leukocytes in the brainstem. Okay, so to circumvent this challenge, we use vagal stimulation. So why we use vagal stimulation? So vagal is the longest cranial nerve and the projects to brain through this nucleus tractus solitarius. And this NTS also projects to LC and other brain regions. So 80% of vagal fiber carry afferent information, which means projects to the brain, the rest projects to your abdomen, okay? So it has been claimed leukocytes modulate brain dynamics mainly through the LC. For example, this study shows if they lesion the LC, the ability of vagal stimulation to suppress seizure is basically attenuated. So also vagal stimulation has been proved by FDA to treat the seizure, to treat depression, and also non-invasive transcutaneous vagal stimulation has been cleared by FDA to treat headache and other disorder like opioid withdrawal, and also in Europe. Okay, so then first thing we like to do is what vagal stimulation really activate LC. So to do this, we also performed experiment in our lab, and in here we implant vagal cuff electrodes around the vagal nerve, and we recorded single unit activity from LC. As you can see, vagal stimulation is able to increase LC firing. Then we performed the same experiment and same analysis to see if back-lobe stimulation is able to improve thalamic information transmission. Turns out it is the case. As you can see in here, back-lobe stimulation increased the feature selectivity and the information transmission by 100%. So more importantly, as back-lobe stimulation suppressed thalamic burst firing, as you may remember, this is similar to direct LC activation. So now we have a spin-off from our lab to commercialize this technology. So we also have some ongoing projects in here. First is we use NeuroPixel to record population neurons in weak behaving animals. So NeuroPixel is really developed core technology. It has 300, technically 1,000 channels. You can simultaneously record from 384 channels. This is what we implanted in our weak behaving animal. And you can see it can easily pick up hundreds neurons. Another project we're doing is to use two-photon calcium imaging to image population activity in the cortex. So before we imaging, we can use intrinsic imaging to map a cortical representation of whiskers. For example, this is our brain vasculature, and this is the intrinsic imaging signal to stimulation of whisker C1, and this is to whisker C2. As you can see, there's some overlap, but there's different. So then we can use our two-photon system to image the calcium signal activity from here. This is our first two-photon imaging. We're very excited about this. We are analyzing the data from these recordings. Okay, so I'm gonna wrap up. I like to thank my lab members and my collaborators. So Charlie here, he did all the neural recording in the LC modulation of thalamic information transmission project. Brian Shriver, he did most behavior training in awake animal and also imaging their pupil. And Cody is doing the NeuroPixel recording, and Steven is doing the two-photon calcium imaging project. And we have a bunch of other projects going. So also like to thank my funding source. Doing animal work expensive without their support and a low way to do this animal work. Thank you very much for your attention, and please email me if you have any questions.